Namaskar, hello, welcome to NCERT's live phone-in program. My name is Tanvi Khurana and like I said, this session is going to be about biology and the topic is Heredity and Evolution Part 1. This is for class 10 students, and if you have any doubts, any queries, you have read this chapter, you want to know anything about heredity and evolution, anything related to this topic, then you can simply send your queries to us on our numbers. You can call us on our numbers and we'll receive your questions and we'll answer them as well. The numbers are 1800112199 and 1800111265. We'll definitely expect your calls, and if you want to send us through email, then the email ID is ciet.kishoremanch@gmail.com. You're watching us live on your YouTube channel at your end. The YouTube channel is NCERT official. You can also write your questions in the live conversation box. We'll receive them instantly and our expert, she'll be answering all those questions in this next 45 minutes, not hours, definitely. So let's meet our expert for this session. We have in our studio Dr. C.V. Shimre. Welcome, ma'am.
Yeah, thank you, Tanvi.
Ma'am is from Department of Education in Science and Mathematics, NCERT. And let's begin with our question answer round. Though I want to ask you, ma'am, last time, if I am not wrong exactly, so we did a session, something similar on the on grounds. So the topic was getting familiarized with some terminologies on heredity, and you explained a few terms. So because these two are related, are these two related?
Oh yeah, of course.
So
of course.
Can you just sum up that session?
Yes, I think I'll just run you through the slides that we saw last session, because these terms are going to be used again in this session, and so I'll just show you or recap some of the definitions or some of the terms. Okay. So let's start with the term heredity. So we had discussed that heredity is a transmission of traits from one generation to the next generation, and heredity is also called inheritance, right? And so the basic or the normal general rule is that the closer you are with your, more closer you are in terms of bloodline, the more similar you're going to look. So the farther you are, you're going to look more different. Then we also discussed about what variation is. So variation, it's different characteristics observed in an individual because of sexual reproduction. And so although we see so many similarities between siblings and all, we also see differences, and that is existing or that is exhibited because of this something called variation that results from sexual reproduction. And we also discuss about trait. So what is this trait? So traits are those characteristics which are transmitted because of the genes, genetically transmitted or genetically which are produced because of the genes. Characters which are produced because of the genes are called traits. And so then we went further to define what are those traits. From where do you get those traits? So traits, based on the definition, every character that you see, you observe in a person, so that includes everything, the height, the color, the hair color, the skin color, the shape, everything. So that consists of the traits. So where do you get these traits from? These traits come from the genes, as we said. So then what is gene? The definition again. So what is gene? So genes are those segments of the chromosome or a DNA, which are units of inheritance. Students have studied in class 9, the definition that they study is that genes are the functional segments of DNA. So we use the term DNA and chromosomes. So then where are these chromosomes and genes? So we come back to that definition again. So what are those chromosomes? The chromosomes are the thread-like structures, and chromosomes appear differently, as students have seen in different books. So sometimes they appear as thread-like structures, but sometimes they also appear in these rod-shaped structures. And so during different stages of cell division, they're able to see different shapes of chromosomes existing in different forms. So then we went further with how the chromosome is uncoiled during cell division and all, before cell division and during cell division, how they divide and all. So then we came up to this stage. So this is how finally the DNA strands look like, the double helix that we see. Then where are the genes here? You know, so then we come back to the definition of genes. So as we said, it is a segment of DNA. So that stretch of DNA, a segment of it consists of genes. So that particular gene, they code for a certain character. So that's how we came about. Then we went further to what chromosomes are, and we discussed that there are 46 chromosomes in human beings. And 46 chromosomes are made up of 22 pairs of autosomes and 2 sex chromosomes. So that's what we discussed. That means 46 are made up of 22 pairs of autosomes and 2 sex chromosomes. That means that there are 22 plus 1 set. So there are 2 sets. So 22 autosomes plus 1 sex chromosome make 1 set. So the another set will be by 22 autosomes and 1 sex chromosome. So there are 2 sets in a diploid cell. That's what we discussed earlier. So a diploid cell will have 2 sets of chromosomes, whereas a haploid cell will have just 1 set of chromosome. And where do you find those diploid cells? You find those diploid cells, all the somatic cells are diploid cells, whereas you find the haploid cells in the gametes. All the gametes, that is the sperms and the eggs, they are haploid. That means they have just 1 set of chromosome. And we went further to, we understood where the genes are. So then we also discussed a few things about genes, you know, like for example, how many genes are there in human beings. So we discussed that the number varies depending upon the researchers. Some researchers came up with the number of 25,000, some going to the extent of 35,000. So it varies, but then it's numerous. I mean, it's huge. Let's say approximately 30,000 genes are present in human beings. So each gene, how are they made up of? Like maybe, again, it varies. So the length varies. So it may be about 27,000 base pairs or something like, so it varies. But the fact is that there are many, many genes in human beings. And for example, we saw this in this particular X chromosomes, there are like 1,098 genes. So similarly, in other chromosomes, there are other genes. We said that there are approximately 30,000 genes. That means that in other remaining chromosomes, there are many, many genes out there. Then we also discussed the term called this one, homologous chromosomes. So what are those homologous chromosomes? So homologous chromosomes, like as you saw in this slide, so they are in pairs, we said. So each pair make up one homolog. It's a homologous chromosome. That means, what does that mean? They mean that they contain genes which code for or which control a particular character. For example, in this particular chromosome, this is a homologous chromosome, a pair of chromosome which is homologous in the sense that they contain gene which code for a particular character. That is here in this case the flower color. Yeah. So they have the same gene that code for a character, that is a flower color. But as you can see in the slide, the genes exist in different forms, different versions. The upper one has allele for purple color flower. The lower one has allele for white flowers. So what are these alleles? Alleles are nothing but the alternate forms of genes, you know. So they are for the same flower color, but they can exist in 2 versions. In this case, purple color or white color. And so when an organism or a plant has this alleles, 2 forms of alleles in this homologous pair, then you say that they are heterozygous. So the organism is heterozygous for that character because there are 2 types, purple and white here. But they can also exist as homozygous. That means the same color allele can be present. For example, both the chromosomes can have allele for purple flower, or both the alleles can have the chromosomes can have alleles for white flowers. So if they have the same kinds of allele, then you'll say that the organism is homozygous for that gene. That means they have the same allele. So that's how. But there are 2 versions in this particular chromosome, and if this organism, in this organism there are 2 versions, that is, it can have purple flower or it can have white flower. So what will be exhibited in that flower, in that plant? So both you cannot have, so you should have either one of this. So what happens is, it has been established that every time this kinds of situation happens, that there are alleles for purple flower and alleles for white flower, then you'll end up having purple flowers, because purple flowers are dominant over the white flowers. So every time you have this heterozygous condition, then you will find that this purple color will be expressed in the organism. That means the purple flower character or the allele is dominant over the white flower allele. So the white flower allele, now we'll call it recessive, because they don't express themselves in the heterozygous condition. Whenever there are two forms, then the recessive allele will not express itself, but it will express itself if both the chromosomes had allele for white flowers. Then you will have the flower in that organism white. Otherwise, you are always going to have, if these two versions exist in an organism, then you are always going to have the purple flower color. So that's in a nutshell. That's really fast, but yeah.
Okay.
Anyway.
Ma'am, I remember you even mentioned two terms, genotype and phenotype.
Yeah.
So can you explain those?
Yes, this genotypes and phenotypes are something that students need to remember again. So genotype is a genetic constitution of the organism. Phenotype is expressed character of the organism. So although the phenotype is also because of the genes or the genetic constitution, but it doesn't tell everything about the genetic constitution. For example, we can still go back to this slide. So if you can see in this slide, we say that when these two alleles are present in an organism, then only the purple flowers will be seen in this plant. So the phenotype is purple color, you know. But we know that there is also an allele or genes for the white flower. So the phenotype do not tell you everything about the genes or the genetic constitution. So we need to understand or go back to the genetic constitution so as to understand how inheritance exactly happens. So that's a basic difference between the genotype and the phenotype. That means that genotype is a genetic constitution, whereas phenotype is expressed one which do not tell you everything about the genes or the genetic constitution. So, yeah.
Okay, okay. Ma'am, you said that we get the genes from our parents, right?
Yeah.
So, and we also get the traits from genes. So, if we need to study the inheritance of the traits, how can we do that?
There are different ways that researchers do. I mean, even Mendel himself did that. So there are a few terms again that we need to understand. For example, students are already familiar with some of these terms. For example, do I have that slide? Yes. So self-pollination is something that they have come across before. They have studied pollination in plants in class 7. So they are quite aware about it. So self-pollination, how does that happen? So when the pollen grains from the same flower, from the one flower, is transferred to the anther of, to the stigma of the same flower, then you call it is a self-pollination. The process is called self-pollination. But when the pollen is transferred from one flower to another flower, stigma of another flower, then you call it cross-pollination. So those are the two basic things that they need to understand, because there is a lot of experiments that's going to happen using this self-pollination and cross-pollination in the process of studying inheritance. Then there is another term they will come across, true breed or pure breed or purebred organisms or flowers, plants, whatever. So how do you get these true breed forms? So in order to get, if I'm interested to get only plants with purple color flower, then first I will choose those flowers among those different kinds of flowers. So the plants which have purple color flower, I will choose those, and I will self-pollinate or I will cross-pollinate with those flowers which have purple color. Okay. You know, I will pick only those plants with purple color flowers. I will either self-pollinate or I will cross-pollinate, but with the same plants which have purple flower colors. Okay. So if I continue doing that for several generations, then what will happen is that I will have only plants which produces purple flower color.
Okay.
Okay, so you do that again and again by picking and choosing. So it is selective, you know. In nature, it doesn't happen that way, but for the purpose of our experiment, in order to study inheritance, that's how we go about. So we pick the particular character that we want. We self-pollinate or cross-pollinate depending upon how we are going about. So after several generations, what's going to happen is that you get the trait of your choice, and that trait will be called true breed, you know, or pure breed.
Okay.
So that's how. So those are the basic processes that is involved in the process of studying inheritance.
Okay. Ma'am, we are talking about humans here, right? So the process is same for the animals too?
Yeah, it's the same. In that sense, in plants, it's much easier. The thing is that because you can just transfer the pollen from one plant to another. So that's very easy. But for human beings, you know how delicate, how complicated it is. So you need to have a sterilized lab, very complicated, very complex, very expensive labs to do that. So it becomes very complicated. So it's much, much easier to do in plants, and especially in some selective plants, it's much easier.
Okay.
You know.
All right. Ma'am, you mentioned Mendel. So can you explain how he went about doing his experiments?
Yeah, yes, Mendel is also called the father of modern genetics.
Okay.
If we look at the background of Mendel a little bit, they have this in the book also. The students can go through their book. So a little bit about Mendel. So Gregor Johann Mendel, he is from Austria. So actually he wanted to be a teacher, and but he failed. But because of his interest in science and mathematics, so he started this kinds of studying about inheritance. So he started growing plants, pea plants selectively, and it was also good that he was good in mathematics. You know, these days everything is interdisciplinary, because if you look at the history of biology, before the time of Mendel or Darwin and all, it was very descriptive. The subject of biology was very descriptive.
Okay.
Descriptive in the sense that it was all about what it is, you know. So how does a flower look like, whether it's tall, what are the characteristics, how many petals it have, how many sepals it have. It was about describing things, not about application or not about predicting, you know.
More theory,
yes,
not practical.
Yes, it was never applied or it was never predictive. So it was more about describing what it is. So but then interestingly, Gregor Mendel had interest in mathematics and science, and so he started applying his knowledge of mathematics in his studies. And so because of his application of mathematics, he was able to come up with different laws of inheritance that we are going to study, discuss about in this session also, a few of those. So that's how Mendel went about. That was a little bit about his background. Then he did a lot of, planting. So he planted a lot of plants. It's not like, okay, I have 5 pots. It's not like that I have a small terrace and so I plant there. No, he had an elaborate field where he planted hundreds and hundreds of pea plants. And why did he choose pea plants? So there are several reasons. Pea plant, it's first of all, it's easy to grow, and it is available in that part of the world. If it was not available, he will not have used it. So it grows, the season completes, it requires a single season to complete its flowering and fruiting and all that. Imagine if he had taken some plants which will flower in 2 years, 3 years, then he will take age as 100, he will require 100 years to come up with all his laws, you know. So pea plant turned out to be very convenient for him. Then he also found that there are very specific contrasting characters in pea plants. He found some 7. For example, pea plants have where the flower is located, whether it's at the tip axial or it's not axial, or whether the seeds are round or wrinkled, whether the pods are constricted or smooth. So the flower colors, you know. So yes, there were specific contrasting characters in the pea plants, which he could identify. So he could pick exactly what he wants, you know. So unless you have different clear-cut differences in those, it's difficult to predict or study further, do your studies. So pea plant was very convenient because it had very specific contrasting characters from which he can pick which characters he wanted to study, how this character is inherited in the next generation. So that made things very easy for him, and so that was the reason he picked. And so he grew a lot of pea plants, and he experimented all of those, and he tried to study what happens if I plant this, if I cross this plant with this plant, a plant with this color and a plant with another color. So what happens if I cross these 2 plants, and what happens in next generation, what happens in next to next generation, you know. He did a lot of studies. So that's what Mendel went about during his studies.
So he reached some conclusion at the end of it?
Definitely, he came up with several things, several laws. The basic laws we are going to study. So I have some slides. For example, if we can see the slide there, if it's clear.
Monohybrid cross.
Yeah, is it clear, or shall we use this one?
It's very clear.
It's clear?
Yeah.
Okay, for me it's not very clear. So, okay, anyway. So, he, first of all, what he did was, he crossed these two plants, pea plants with purple flower color and pea plants with white flower colors. Students should remember that when we say they crossed these two, he crossed these two plants with different flower colors, it doesn't just happen that these two cross each other and they form a new plant. It doesn't happen so easily. It's much complicated. They must understand that when we say that these two are crossed, they must go back to the process of fertilization or pollination. That means that the pollen from purple flower color was transferred to the stigma of white flower colors, white flowers.
Okay.
Or the pollen from white flowers were transferred to the stigma of the purple flower, you know. So that's how it's going to be. So what does the pollen contains? So it contains the gametes again, you know. So that's how they should understand. So it's ultimately what's going to happen is that the gametes are going to meet. So first of all, they have to identify the gametes. What kinds of gametes will be present in the white flowers? What kind of gametes will be present in these purple flowers? So once you transfer those pollen from one plant to another or one flower to another, then there is mixing of those gametes. So what do you have? So when the gametes form, the zygote is formed. From the zygote you come across the fruits, or in the case of humans being, a new human being is formed, like, you know. So that happens. But in case of plants, the flower will produce fruits, right?
Right.
After fertilization, the flowers will produce fruits. You will grow the fruits. Then you will find that what kind of flower is produced by that fruit.
Right.
You know, so in the diagram, if we can go back to the diagram, can we go back to the diagram, previous slide? So in that slide, if you can see there, so it seems that it's so easy, you know, as if like you cross this purple flower color and white color flower, and then bam, it comes out the purple flower color. It doesn't happen that way. So it has, the gametes has to mix, then the fruits are formed, the fruits are grown, then what kind of flower is produced. And he found that when he crossed these two flowers, he found that all the progeny, all the individuals, all the plants bore purple flower color.
Okay.
What is important here is that they should understand the words there at the top on the left hand side, just below the P generation in the slide. Can we go back to the slide, please? Yes, just below the P generation, the word true breeding is written there, right?
True breeding.
True breeding, true breed. I use a word, I explained the term true breed before. What is true breed? That means that it is going to give rise only to certain character. For example, if a purple flower is a true breed here, so it is pure. That means that it's going to give rise only to purple color flower. It's not going to give rise in any stage to white flower. Flowers. Similarly, when I say white flower is true bred, or it's a true breeding, that means that the white flower plant is going to give rise only to white plants which have white flowers. So when he did this experiment of breeding or crossing two plants with true breeding nature, that means one plant had a true bred for the purple flower, another plant is true bred for white flower. So when he crossed these two, he found that all the offsprings in the next generation had purple flower color. Okay. Then now it's a hybrid now, right? So so it's a hybrid that has formed. For example, because we will go back to the next slide. Is this slide visible, or shall I use the overhead camera?
Not really.
So it's not visible there? Or shall I use this overhead camera? Is this better? Whichever is better. So,
ma'am, just explain it.
Okay, without looking at the diagrams.
Now that we have shown both of them, I'm sure the audience will understand. Yeah.
So it's like, so they have it. So if we go into the genetics of that, the genotype of that, so if you can see there in the slide, can we go back to the screen, the overhead camera? Yes. So on the top, you see a P generation. The P generation has like purple flower and the white flower.
Right.
So just below the purple flower, you see purple flowers and 2 P's in big capital letters, 2 P's there. That is the genotype.
Okay.
The 2 P's are the genotype. The phenotype is a purple color flower, right?
Okay.
So the genotype is a P and P. That is the genetic constitution. Whereas just below the white flower color, you see 2 small P's there.
Yeah.
So that is the genotype of this white flower. Whereas the phenotype of this white flower is white color.
Okay.
So that's a phenotype, but the genotype is a 2 small P's there. So when he crossed these 2, that is 2 big P's and 2 small P's. So what kind of gametes this individuals will produce? So the purple flowers, plants with purple flowers will produce gametes of, like, if you can see there in this slide, the P there, I'll just show this. So the P here,
that's
with
a capital
circle,
one.
capital and circle, because that is the gametes. It represents a gamete.
Okay.
So the purple flower color will produce only this kind of gametes with capital P.
Okay.
Whereas the white flower colors will produce gametes only of these kinds of gametes.
Small
p. You know, and so when this is crossbreed, I mean cross-fertilized, when there is pollination happening between these 2 plants, so basically, as I said, it is not the flowers that is merging, it is the gametes that's merging. And ultimately, when these 2 gametes merge, so you find 1 big P and one small p in the next generation. That means
Okay.
an individual, a zygote with big P and small p will be formed. That zygote will grow into seed. The seed will be planted. After it is planted, the seed will produce flowers of purple flower colors. But the purple flower colors will be now 1 big P and 1 small p, because you can see the gametes there. It is 1 big P and the 1 small p there. So when it is hybridized, so you find purple flower color still, the phenotype is still purple flower color, but now the genotype is 1 big P and 1 small p. So that's the difference. So they can compare the difference between a pure breed there, where you have big 2 P's, whereas in a hybrid, you have 1 big P and 1 small p, but still the phenotype is purple flower color.
So if the white flower would have been the pure breed, then the result would have been again 1 big P and 1 small p, but the flower would be white flower.
No, for in case of, as I said, in homozygous, the recessive genes, for example, this is the recessive, it has this flower with white flower has a recessive allele. That means that it will only express when there are 2 small p's, only in a homozygous condition.
Okay.
Go back to the, if you recall the homozygous definition and the heterozygous condition, whenever there are 2 alleles, 2 versions, the dominant character will be expressed.
Okay.
Whereas whenever they're in a homozygous condition, the recessive condition can also be expressed. For example, in this case, it is white flower because you have the genotypic constitution as small p and small p, that is in a homozygous recessive condition. That is the reason you are finding it, you're finding white flower here. But here in this case, in this F1 generation, that means first filial generation, you have a hybrid now. A hybrid, one big P coming from this gamete, 1 small p coming from this gamete, and so the individual gives rise to flowers which bear purple color. But the genetic constitution is a hybrid, 1 big P and 1 small p. And so monohybrid cross, that means monohybrid cross is nothing but a cross when individuals with 2 heterozygous characters are crossed. So you are going to cross this individual, these plants which have 1 big P and 1 small p, that is in heterozygous condition, with another plant of this type. So you are going to cross these 2 with two hybrids with in heterozygous condition. So when you cross, when you take this plant, 2 plants of this type and cross them, what you're going to find is this. You know, so it's the same. So the gametes that you can that can be produced from this kind of flower is 1 big P and 1 small p. So half of all the gametes will be big P, half of all the gametes will be small p. So 2 types of gametes can be formed here, 1 big P and small p types. So since you are taking a crossing two of these types, so you can keep them in this. So you can just assign one of those flowers as male and one of those flowers as female. So then the kinds of gametes that can be formed here is in both the plants, because they're the same, you are crossing the same kinds of flowers. And so you have gamete one big P, gamete with one small p here as representing as a sperm. On the other side you have gamete with one big P and gamete with one small p representing the eggs gametes. So when these two combine, these two fuses, the big P's, the gamete with big P and the gamete with big P, when they fuse, then they produce purple flower color with two big P's.
Okay.
Right? Here it is a small p here. Here it is a big P. Here it's a big P. So what happens? It will produce flowers, plants with
Purple color.
Purple color, but the genetic constitution will be one big P and one small p, because the gametes are, you know, one small p and one big P. So you have this kinds of genetic constitution, but still the flower color is purple. Similarly, in this case, you have this small p and hybridization of the small p. So what happens is, here in this case it is a big P, but here if you can look here, one big P and one small p, so it produces a genetic constitution is one big P and one small p, so it will still bear purple flower color. But here in the last slide, in the last corner, what happens is hybridization of the gametes with small p and small p is happening, and that is the reason now you are going to have flower with white color. Because now the genetic constitution is in a homozygous recessive condition. That is the reason you have this white flowers. So in other three cases, what's happening is you have purple color flowers but different genetic constitution. Right? So sometimes it is two big P's, sometimes it is one big P and one small p. So the phenotype is purple color, but the genotypes are different. So that's how. So the difference between genotype and phenotype, it's different. Phenotype comes because of genetic constitution, but it does not tell you everything about the genetic constitution. So genetic constitution can be understood only by understanding the details of the into this, by going into that. So what happens is that, so you can say that in the F2 generation, filial 2 generation, you find in this type of cross, you find the phenotypic characters. Typically you'll find 3:1 ratio. That means every time you cross this type of plants, this type of monohybrid cross, if you do that, you'll come across a ratio of 3:1 phenotypic ratio.
Always.
Always.
Okay.
But when we say 3:1, it is not exactly 3:1. I have a slide, I think. Okay. For example, there you can see the slide there. So it is like 3.15 is to 1. So it is something like that. So it is not exact, approximately it is going to be 3:1, right? So Mendel did this experiment for other characteristics also. For example, you have there the flower position there, terminal or axial. Then he did for the seed color also, similarly. Then he did for seed shape, he did for the seed pod shape, the cover. Then he did for all 7 characteristics, 7 contrasting characteristics. But if you can see the last corner column, you'll find that, yes, it's almost, 3:1 always. Sometimes it's about a little bit over 3:1. Sometimes it's a little bit below 3:1. But approximately,
in this case, it's 2.84
yes,
to 1.
Yeah, you can still say that it is, because since it is 2.8, you can still round up the figure and say it's 3. So right. It's like, so you're going to get approximately 3:1 phenotypic ratio.
Okay.
But if you go back to that slide again here, if you see the genotypic ratio, you'll find that the genotypes are different for these 3 purple flower colors. Right? For one, it's totally different, big P and big P, whereas the other 2 has same genetic constitution here. 1 big P and 1 small p, 1 big P and 1 small p, right? So you will say that the genotypic ratio will be 1:2:1, because you have 1 of this type, 2 of this type, and 1 of this type. So the phenotypic ratio will be 3:1, whereas the genotypic ratio will be 1:2:1. So that's how he came, finally came across that. So then what what happens after that? So as I said, he tried with all kinds of characteristics in all these different contrasting characters. So what did he find? So the findings. So it's interesting to see that. So students can just go through that slide. So first, what did he find? There are alternative versions exist in genes occur in individuals. You know, inherited characters are different because of the presence of different versions of the genes. So that's the first finding that he found. He also found that for each character, an individual gets a copy of the gene from one parent, another copy from another parent. Then the third conclusion is that these two genes, if these two genes at the locus are different. For example, in that case, we studied about the purple color flower and the white color. So if these two exist, then one will be always dominant, one will be recessive. You know, that's what he concluded. Then the last thing is that he found out that these two alleles will never be come together in a gamete. So they will always segregate or separate during the formation of the gamete. For example, we say that an individual with big P and small p, even if they that is the genetic constitution, when they form gametes, it is going to be one small p or one big P. It's never going to be both. So these two alleles, they segregate during the formation of gametes. That's what he concluded from his experiments. Right. So that's about the monohybrid cross. So do we still have time for the hybrid cross?
We have got 4 minutes. But I wanted to ask, so this conclusion came when he took one character.
Yes.
He contrasted one contrasting character. But if he takes two contrasting characters?
Yes, so he was interested to find out. So when it is a pure breed, it's easy, you know, it's easy to predict. Because in a pure breed, you know that, in a true breed, you know that the individuals, the offsprings are going to be of that character. But when it is in a hybrid nature or in a heterozygous, if the organism has alleles which are heterozygous for the gene, then how it's going to be inherited?
Right.
So that is a question that he had. So he deduced that for one contrasting character, for the one monohybrid cross, he did that. So he did that for two characters also, similarly. So he did similar characteristic, cross. For example, you can see the slide there. So maybe I think we should take this up in the next session because it's going to take a little bit of time. So, but I'll just tell them what basically he did. Yeah. So he took two characteristics in this case. For example, he took the seed color. So in pea, plant students might also be aware, they have, sometimes it's green color. There are also a variety of brown color seeds, right? Have you come across the pea plant, pea seeds? Do you know what a pea seed is?
Yeah, I do.
Yeah. So you have green pea and brown pea.
Okay.
Are you aware of this? So
No.
you should go back to a grocery and check that. So there are brown peas and green peas. So you will just call those brown peas as yellow peas. So he took a characteristic of this pea color, and he took another characteristic of whether it was wrinkled, the shape.
Okay.
Sometimes this pea will be round, totally round. Sometimes it will be wrinkled, you know, it will not be smooth. So he took those two characteristics, the color and the shape. So he took those two characters to study how the hybrid will be inherited in the next generation. So that's what he did for the dihybrid cross. So since that is going to take a little bit time, we will not get into that.
Sure.
So
now
we'll stop at that.
You've touched the basics.
Yes.
We'll continue it in the other class, part 2 version of heredity and evolution. But before leaving, do you want to give any activity to our students like you gave me, go to the grocery store?
Okay, I have actually an activity there.
Sure.
So this is from a textbook. So if students who are not from using NCERT books, they can read this line, this slide. So this slide tells them about, they should look for, can you just read that? Observe
the ears of all the students in the class. Prepare a list of students having free or attached earlobes and calculate the percentage of students having each, figure 9.2. Find out about the earlobes of the parents of each student in the class. Correlate the earlobe type of each student with that of their parents. Based on this evidence, suggest a possible rule for the inheritance of earlobe types.
Okay, since they're not going to school, what they can do is they can look at the earlobes of all their relatives who are in and around their house, in their family, you know. So they can see the earlobes, whether the earlobes are attached or they are free.
Okay.
Some earlobes will be attached to the skin, to the face, or to the side of this. Some earlobes will be free. So how many people in their family or in their close relatives have free earlobes or attached earlobes? They can then just calculate and see the interesting results that come out.
Sure,
right?
that's a good observation. Thank you. Thank you so much for being here and teaching us about heredity and evolution, though we are taking up this in the next class as well.
Yeah.
Thank you for all of you for watching this session. I'm sure this was very useful for all of you, for all the class 10 students. But don't go anywhere because we're coming back in a short while and that session will be on the topic स्वतंत्रता की ओर. हिंदी का एक सेशन हम थोड़ी देर में वापस आते हैं लेकर और ये होगा कक्षा चौथी के बच्चों के लिए. तो सभी बच्चे, पेरेंट्स और टीचर्स, आप सभी से रिक्वेस्ट है कि अपने बच्चों के साथ रहिए और स्टूडेंट्स प्लीज कॉन्सेंट्रेट ऑन योर स्टडीज. दिस इज अ वेरी प्रेशियस टाइम फॉर ऑल ऑफ यू. थैंक यू सो मच. स्टे हेल्दी, स्टे स्माइली. नमस्ते.